Welcome to Science Wars, Chemistry, Bio, Physics, Medical, which field is best? Understand the force and energy, quantum mechanics and relativity. My field is pure and they rely on...
Hello, in this video we are going to do the virtual lab for fermentation. The reason we're doing a video is that it is an Adobe document and sometimes it does not open on your computers, and so we're just making a video of it. And as we go through the video, you will fill out this virtual fermentation lab document. That is what you downloaded. So let's get started on this. So this is the website and you should have your document opened up and ready to go. So let's go ahead and push start. And these are the materials that we're going to need for this. You do not need to write any of these down. So we're going to need water, glucose, which is a sweet energy substance, yeast, which is a living material. You might know it from bread. We use yeast to make bread, or to make the bread rise. Actually, it produces a gas that makes the bread rise. And flour, which can also be used in many cooking things, including bread. In addition, the equipment we're going to use is a heater, 5 flasks, and a bunch of balloons, 5 to be exact. So now we need to start recording some information. So in flask 1, we have water and yeast. So over here, you would go ahead and type in water and yeast. Next, in flask 2, we have water, yeast, and glucose. In flask 3, we have water, yeast, and glucose, but then we put it on a heater and we heat it for 3 minutes, which basically brings it to a boil. So we boil it. So water, yeast, glucose, and we boil it. In flask 4, we have water and glucose. In flask 5, we have water, yeast, and flour. Okay, so those are our ingredients. So next thing we're going to do is we are going to go ahead and put a balloon on these, and we're going to leave them. So leave for fermentation to take place for 24 hours. We are going to measure how much gas is going to come out of these flasks. Okay, so we're going to leave them for 24 hours and see what happens. And again, what is going on here is we've capped these, so they will use up some of the oxygen that's in here, but then the oxygen will run out and they'll start doing fermentation, which is where they are breaking down a food source and producing a gas, right? So which one of these do you think would grow the most? Well, it turns out that 3 of them did not grow and 2 of them did. So what we're going to do is we're going to go ahead and see which one's right and which one isn't right. So what you can do right now is go ahead and write your claim right here on if fermentation took place or not. So if it took place, it means that there's gas in the balloon. So you're going to go ahead and write here yes or no, did it take place? So do you think water and yeast fermented? Do you think it gave off a gas? So you'll write either yes, or if you think it's the other way around, you'll write no. So take a second, go ahead and fill these in, and then we'll go ahead and start the video, and we're going to see what the results were. Alright, so let's say you think that water and yeast, alright, it sounds would make something. We have yeast and water together, so maybe that fermented. So let's go ahead and, ooh, no, that didn't work. Alright, so maybe it made a lot of fermentation. No, that's not it. Well, that must mean that it didn't grow at all. So yes, for water and yeast, no was the answer. It did not ferment. It did not make any gas. Okay. Well, what about yeast, flour, and water? Or what about yeast, glucose, and water? Well, one of these, or both of them, probably worked. But we're going to go ahead and do the other ones just to see. So glucose and water. Again, here there is no yeast at all. So if there's no yeast, would it work? No. No. So without yeast, again, there's nothing to break down the glucose, nothing to give off a gas. So of course it's not going to work. Well, what happens if we boiled the water? Well, if we boiled the water, we find out that nothing happens. And you'll find out later that the boiling of the water actually kills the yeast. If the yeast is killed, then the glucose and water the glucose can't be broken down because the yeast is dead. So the next question is, well, which one would it break down and grow the best in, flour or glucose? All right, well, let's say that maybe it's the flour. Flour is a pretty high energy food, but no, that is not correct. It turns out that flour is a very complex molecule, and it's made up of glucose. So for the yeast to get the energy in flour, it has to break it down to another material to get it to glucose. So this is just a faster way for the yeast to get the energy, so it grows more because it doesn't have to take as long. Here you have to break the flour down to get to the glucose, so it's not going to grow as fast. So from your results, you should have that flask 2, yeast, water, and glucose, fermentation happened, and in flask 5, yeast, water, and flour, it happened. So now we're going to find out a little bit more evidence about this. So we're going to continue. We're going to see if we can find out what's going to go on with the gas. So what we do is we take flask 2 and flask 5 and we push the gas through just regular water. So we're dissolving the gas from the balloons into regular water for these two flasks, 2 and 5. And then what we're going to do is we're going to do a litmus test. So we're taking red litmus paper, and red litmus paper will stay red in an acidic solution, and it'll turn blue in a basic solution, right? So we're supposed to guess which one it is. Well, let's say, and it turns out that this part isn't working, but if we pick this, you'd find out that no, it's not alkaline because it does not turn blue. It actually does this, and unfortunately this part isn't working right now, but it would turn. So let's go to our next one. And then we would take an alkaline strip, a blue litmus paper, which is an alkaline strip, and we would see what would happen. And if it was alkaline, it would stay blue, and if it was acidic, it would turn red. So let's go to our results. We'll see that in our results, the red litmus paper stayed red, which means it was acidic. Right, but that could also mean it was neutral. It did not turn blue, so we knew it was one of these two. So we took our alkaline strip and we put it in there, and the alkaline strip turned red, which means it has to be acidic because it stays blue in neutral and it stays blue here. So we know that because this one turned red and this one turned red, or this one stayed red and this one turned red, that we have an acidic solution. So dissolved gas turned water into an acidic solution. Do you have any ideas on what the gas is being produced during fermentation? And the answer is carbon dioxide. So going back, we're going to go ahead and look at some evidence here for what was happening. So evidence for what was happening. So we have no gas collected in the balloon, no glucose as food for fermentation to place. There is no CO2 is produced. So over here, we can go ahead and write some of this evidence down. So fermentation in this case did not happen because it was a no. And the evidence for that was there was no gas in the balloon. Reasoning behind that would be, to explain why, there is no glucose as food for fermentation, right? And therefore no gas was produced. So no carbon dioxide gas was produced during fermentation because there's no food. We can go to the next one. So after 24 hours with the glucose, the yeast was able to break down the glucose easily. It produced a lot of gas, and it made the biggest balloon. So here, we definitely had fermentation. So in here, this should have been a yes, right? And then the reason why is largest balloon, most gas caught. Balloon was the most full of gas. As yeast utilized the glucose, it produced CO2. Utilized or broke down. So the balloon was the most full of gas, or was the fullest of gas. I don't think my word likes that, my word choice there. And actually that would be the evidence. This would be the reasoning over here. So balloon was most full of gas, so this actually is our reasoning over here. So I'm not going to type any more of these through. I'll let you guys go ahead and do that. I will play the evidence and the reasoning for each one of these, and you should have your claim, evidence, reasoning set. So let's go to our next one. So this is flask 3. In flask 3 we had a yeast, glucose, and water boiling, and the balloon did not inflate, so no fermentation took place. We know this, the evidence was that no gas was collected in the balloon, and the reasoning is that boiling of the water probably killed the yeast, so they were not able to ferment, and thus no CO2 was produced. In flask 4 we had glucose and water, and with just glucose and water, right, no balloon was made. And again, this is a nice control to make sure that it wasn't the glucose that was making the balloon expand, that it was the yeast. So here, no yeast to break down the glucose. So no gas was collected in the balloon. That was our evidence. And the reason is no yeast for fermentation to take place. So no gas was produced. And finally, in our flask 5, we did have gas collected over 24 hours. It just wasn't the most gas. Gas collected was less than that collected in flask 2. So in flask 2, again, glucose is an easier thing to break down. Flour, it's more complex, requires more time to break it down, so less energy was able to be used for growing and reproducing. So the evidence was, again, less full. The reasoning, as flour is a mixture of complex carbohydrates, yeast took longer to break down the flour into glucose, that being used for fermentation. All right, so that is our evidence reasoning. And this is the principle that you're going to be looked at later, so make sure you sort of understand this. Yeast contains an enzyme called zymase. Zymase catalyzes the breakdown of glucose to produce ethanol and carbon dioxide. So again, one of the questions that's going to come up is, does yeast do aerobic respiration? They do. But in the case that we had, the oxygen was quickly used up, and so they had to go to fermentation. In fermentation, they use this enzyme zymase to break down glucose, all right? And in fermentation, they make carbon dioxide and ethanol. Okay, then what happens is that CO2 will interact with water, and it'll make an acid. Well, this carbonic acid has it disbonds with the H, and so it turns the water acidic, which is why the litmus paper turned red, and why the alkaline also turned red, showing that it was an acid. So again, this is a principle that's going to come up later more with the acidification of oceans, which is if you have a lot of carbon dioxide, it can turn water acidic, which is going to be a problem for the oceans, for coral, for aquatic creatures, etc. And we can see that in this lab here, where we're just showing, hey, CO2 does turn water acidic, and that's how we knew that it was CO2, because the gas had to be CO2 because it turned the water acidic. Right, and that concludes this part of the lab. Let's go to our document and make sure everything is answered. So you will have all this filled out now. Then you go down to your conclusion questions here. Why do yeast use fermentation in anaerobic respiration rather than aerobic? And again, it isn't that they aren't using aerobic, they were. It's just that the oxygen ran out in the aerobic respiration, and they had to switch to anaerobic, which is a process where you're breaking down glucose and you're making alcohol, and you're making CO2 gas. Okay, are yeast autotrophs or heterotrophs? What do they use for energy? What activities might yeast use energy for? Okay, so I'm going to let you just think about this. You can Google these answers if you need to, but you should have kind of an idea based on what is going on in this lab. All right, if you have any further questions, please ask me, your science teacher.